Thank you very much, Mickey. It's really a great pleasure to be here, especially to visit the home of Avram Hershko, who I first met actually in Gordy Tompkins' lab must have been the summer of 1970, possibly 69, probably 70 actually. And as Avram said, Gordy was an absolutely magnetic personality. He was one of the very few people I know who could actually make biochemistry funny. And I'm going to try and do the same for you now, but I should warn you, this is actually a scientific talk. And I'm going to try and explain what we know and how we find it out. And I'm a little nervous, to tell the truth, because I had to give a talk, of all places, in the British Embassy in Paris. And the British ambassador, his wife entertained my family and me to breakfast. The British ambassador had to take the first train to London, and he came back in time for my talk. That's how good the train service between Paris and London is these days. And after my talk, he said, well, that was very interesting, he said, but I didn't understand a word of it. And I was a little downhearted because it was the same kind of talk that I give to my children in the 7-year-old school. So we'll see. So I may be overestimating your intelligence, but I don't think so. So this is the title of the talk is Getting In and Out of Mitosis, but I really want to, you'll see I'm very interested in switches, when things change. Here is a switch. Some of you may recognize it. And behind this switch, it's not quite so simple. It has a lot of parts, and it's kind of hard if you're mechanically challenged, as I am, to figure out what all those little springs exactly are for, and if you take it apart, it'll probably fly into bits and you won't be able to put it back together again. And our aim is to understand biological processes in a similar way. So we'll also be talking about nanotechnology, and I'll show you that. So here's a typical transformation. Bottom, you may remember, is under a spell, and it's given him an ass's head. Later on, the spell is broken, he comes back to human form. And I show this because this is my first public appearance, and here am I. And during this performance, I wasn't allowed to witness the love scene between Titania and Bottom because the director said I had to point the other way. And I was a very naughty little boy, and on the final night, when the director couldn't give any sanctions, I turned around, so I watched what was going on. And he noticed. So what I'm going to talk about is the most fundamental thing, really, in biology, which is one cell becoming two cells. And one of the things that I believe, and it is a belief rather than a proven fact, is that we are all descended from the same cell. Every one of us in this audience. Not only are we all descended from the same cell, but so this is our cousin. And the more we sequence DNA, the more we discover that actually, you know, we're terribly close to these plants, actually, in sort of biological space. And as far as I know, cells only ever come from other cells, and you can trace your descent back to an original cell on Earth sometime between about a billion and 2 billion years ago. So, and this process of cell division has been happening ever since, and look where it's got us. So this is it. This is a fertilized sea urchin egg, and it's going to divide, and you can see that nothing very much, it's not a terribly dramatic process until the very end. And then it's so beautiful and majestic. This, of course, is very much speeded up. But that division in two cells is what I'm going to try to explain to you now. How does it happen? What's going on inside? Well, there are preparations for mitosis, and then those preparations have to be undone to get out of mitosis. And the most important thing, and it's really confusing, you know, you should never have lectures with two screens because you can't point to either of them. So I'll point to this one on the left, and excuse me on the right. So when cells go into mitosis, the most dramatic thing, and some of you might have noticed in the movie, I won't show it again because of time, is that the cell contains the nucleus where the genetic material is, and the most important thing there is the DNA and the chromosomes and the proteins that packages them up. And the nuclear envelope dissolves. I'll show you that in slightly more detail in just a second. And then the chromosomes are lined up, and already there are two chromosomes which are direct daughters of one another, exact copies, and those two chromosomes will then, here they are again, at the climax of mitosis, are pulled apart and sent to opposite daughter cells. And that's how cell inheritance works. The information and how to make a human being is all contained in these chromosomes, or how to make a gladiolus is contained in those chromosomes. And the whole point of cell division is to distribute the DNA molecules, the chromosomes, accurately to each daughter cells. And when that goes wrong and a cell has missing chromosomes, that's what gives rise to cancer. That's why people consider the process of cell division fundamental to the cancer process. So I'm interested in what it is that switches this process on, and then what happens when it's switched off. How is it all controlled? And it's a complicated process, and as you can see from the previous, it's a diagram. So this was what was known in 1961. Dan Mazier was a kind of prophet-like professor at Berkeley, and he invented this concept, which later in life he said hadn't proved terribly useful, but I think it's very useful, the starting pistol. Because everything comes up to the moment when the chromosomes are lined up in the center of the cell, and then if you watch a movie of the process, exactly as though somebody fired a starting pistol and the chromosomes start moving apart. And then lots of things have to happen. You can say, don't worry about the details. All kinds of complicated things have to happen before and have to happen after. But we're going to really focus on this pistol to begin with. So here is a cell just 15 minutes apart. This happens to be a clam egg. And you can see it's got this beautiful nucleus. If you could see the chromosomes, they will all be lined up around the inside of the nuclear envelope here. And here you can just about see them lined up in metaphase. And you can see that between here and here, something very dramatic has happened. This beautiful structure that you see before fertilization is completely reorganized. The nuclear envelope is broken down. And there is some kind of structure you can dimly make out here, which was clearly diagrammed in the thing. We can see it more clearly here. This is called the mitotic spindle. This is the thing that lines the chromosomes up. And it's made of protein. It's a little nano machine designed to line chromosomes up and then pull them apart. And apart from the chromosomes and the microtubules that join the chromosomes to these poles, there are the poles and there are the little connectors, which you probably can't see. I can't see from here. There are little red dots here which connect these tubes to the poles. And basically, it's very simple. You pull on the string and the chromosome moves apart to the pole. And then finally, the cell divides into two. And if you think about it for a moment, you realize that this plane of division here is extremely important. You want the two sets of chromosomes parting to be here and here. You don't want them to be here and here, because that would be a total disaster. So there's a lot of very fancy nanoengineering going on in these cells to organize this process. So how do we know about its control? Well, the answer started out from studying reproductive physiology in frogs. Not a very promising beginning. And that was really all the pioneers were interested in. And they discovered that the hormone progesterone was what caused this change. And this movie, you show this white spot appear. This is a frog oocyte. That's the cell which will become an egg. You find it inside females, in female frogs in the spring. And these cells, they're very large, about the size of a match head, will pass down the oviduct of the female to be fertilized with the sperm when they come out at the back end of the female. Okay, and progesterone causes the appearance of this white spot. What else does it do? Well, if you look underneath this white spot, what you find is chromosomes here, fluorescent blue, lined up on what looks like a spindle. And with this kind of microscopy, you can't tell what it's made of, but these are little protein threads joined at a pole here, lining up the chromosomes. And what a Japanese, Yoshio Mizui, working in Clem Market's lab in Yale, discovered was that what progesterone did was to turn on an enzyme. And it was an exceedingly interesting enzyme. It was an enzyme that actually catalyzed cell division. When I first heard about this, I thought it was an extraordinary thing, because you know about enzymes that will degrade things. Here was an enzyme that did something immensely constructive. It actually caused a biological switch to make the cells divide. They're poised on the edge of division. This stuff turns on, and then it catalyzes the breakdown of the nuclear envelope, the alignment of the chromosomes, the assembly of this complicated spindle thing, and then the separation of the chromosomes to the two daughter cells. And not only was this, it turned out this was a protein. They knew that because if they boiled it, it died. And if they treated it with proteolytic enzymes, it died. And it had the amazing property of turning itself on. A little bit, there was some kind of positive feedback loop which turned more of it on once it had started to be made. So it was really, really interesting. And I first heard about this in 1979 from a man called John Gerhardt. And I thought, boy, this would be fun to work on. But it wasn't what I was working on, so I didn't. And it turned out to be absolutely fundamental. So what is it? Well, people, Pache Avram's talk, tried to purify, and it turned out to be exceedingly difficult to purify. And really, they didn't succeed for about 15 years. And it wasn't the pioneers who finally succeeded in purifying it. But along the way, its properties were very well defined. And it turned out this stuff, which they called maturation-promoting factor, because they didn't really know what it was, or indeed what it did in molecular terms. This maturation-promoting factor really was very interesting, because every time the cells divided, every time you could see the chromosomes and the nucleus was broken down, the levels of this factor were high inside the cells. And every time in between, when the DNA was being replicated and the cells were just growing, you couldn't detect MPF. So it really was, you only found it in dividing cells. Now, initially, it was only found in frog eggs when they were maturing, but then somebody looked after fertilization and found that MPF reappeared after fertilization, again, every time the cells were dividing. So it's pretty interesting stuff. It bounces up and down like a yo-yo as the cells divide. Not only was it found in frogs, but then a group started looking in starfish. Now, starfish are quite a long way away from frogs, evolutionarily speaking, and I'm not sure if they're kosher or not, actually. They don't have, they probably aren't, whereas the frogs are, I guess. Anyway, starfish turned out to have this stuff too, and in starfish it bounced up and down with the cell cycle, and amazingly, starfish MPF worked in frogs. Not only did starfish MPF work in frogs, but also it later turned out, about a decade later, that you could find MPF, which worked in frogs, in human cells when they were dividing. So suddenly, from being a rather curious phenomenon related only to frog reproduction, this stuff took center stage and was clearly something very important, intimately connected with the control of cell division. And this, so to speak, is where I came in. I was actually interested in the control of protein synthesis in sea urchin eggs. And one summer's day in 1982, I did this very simple experiment, which is kind of complicated to explain for those of you who have never run a one-dimensional SDS polyacrylamide gel. But basically, if you see this dark band here, I don't know if, let's see if you can see it. I'm closer to this one over here. Let's switch sides, huh? That's a protein, a newly synthesized protein. And what you can see is it comes up and then it goes away, and then it comes back again and then it goes away again. In other words, this protein came and went in time with the cell division cycle in a way that was extraordinarily reminiscent of that up and down cyclical behavior of MPF. So being a cautious and skeptical scientist, I realized that I had made a great discovery because it went away. That was, and people, actually it's very hard to explain. See, people say, what did you win the Nobel Prize for? And I say, I discovered something that went away. It's not very dramatic. It's really not terribly interesting, actually. And I was terrifically excited by this, and the question was, what was the relationship between this behavior, coming and going, and the control of the cell cycle? And then later that summer we looked in clams and we saw not one but two of these proteins. It's rather clearer here, I think, the coming and going. And these are called cyclin A and cyclin B. Why are they called cyclins? Because I was a very keen cyclist at the time. It was a jokey name. and it also because they go up and down in time with the cell cycle. And it was very easy to show that if you blocked the cell division cycling, for example, by inhibiting chromosome replication, then these cyclins did not go away. They only went away if the cells actually entered mitosis. And so we still don't actually know, and we're still working on why it is that cyclin A, you can see, goes away before cyclin B. But believe me, these two proteins are extremely important in controlling the cell division cycle. And their real importance was proven by a former student of mine who then did a very distinguished PhD and went to do a postdoc with Mark Kirscher, and I'm still good friends with Andrew. He actually showed that all you had to do to make a frog egg undergo a cell division cycle was to make this protein cyclin, and that it accumulated. Then you can actually see the cells enter mitosis by this little shift, of which more in just a second, and then it goes away. And Andrew also showed that if the protein didn't go away, which he could do by making mutations in the proteins, then the cells were actually locked in the climax of mitosis forever and ever. And actually what normally happens is that the cells would then die. So you have to make it in order for the cells to enter mitosis, and you have to destroy it in order for cells to get out the other side. And it took a long time, Mark Kirschner and his associates and Avram Hershko, together with Joan Ruderman, showed that actually the mechanism of destruction here was the mechanism that they had discovered in their beautiful series of experiments using the ubiquitin degradation system. And a lot is now known about the control of that, but we're still working on it, and it's still not completely clear how it's controlled. And then in collaboration with the yeast geneticists, I mean not really collaboration, but in a sort of parallel universe, it turned out that the cyclins here shown by its yeast genetic name, CDC13, combined with Paul Nurse's and Lee Hartwell's protein CDC2, and actually MPF was made of two components, one of which was completely constant and the other of which came and went. And I'm now going to show you a quantum mechanical movie showing you how this nanomachine works. This is CDC2 twiddling round and round, and cyclin is going to come in from the right. And what cyclin does is actually to pull a loop of protein away, out of the way, so that the enzyme can work, and it turns it on. But it's really a very complicated little process, because not only that, in addition to that, we have to stick a phosphate on that residue here, and you'll see the phosphate come in shortly. Plop! And then in comes the substrate from the left, and this phosphate group, it's all about phosphates, this talk, is going to be attacked by that serine residue, and that little peptide here, a little model protein, is going to have a phosphate stuck on it, and it'll go away, and then the whole process can repeat itself, and this enzyme can work, not terribly fast, about 3 times a second, something like that, to stick phosphate groups onto other proteins. It looks like this. Here's a target protein. Here's the protein kinase. We've just seen a protein kinase. To turn this thing on, you have to stick a phosphate group on it. And to turn it off, you have to take the phosphate group off. It's really very, very simple. But in practice, in real life, if you actually look at this, here's our previous. This is the protein kinase that does it, which I said was turned on by phosphorylation. This is so dim I can't even see. I mean, you know, oh, there we go. That's better. There's the phosphorylated residue. There it is before phosphate. You can see it's really rather a small shift. This thing just gets pulled up to there. But that results, amazingly, in a 100-fold activation of this enzyme. So sometimes one phosphate can make an enormous difference to the activity of the enzyme. I mean, it's catalyzed by a protein kinase. And obviously, because that is put on by a protein kinase, to turn this thing off, you have to remove that phosphate. Or there's another way, which is to degrade that cyclin subunit on the right there. So this is a complicated enzyme. It can be turned on by phosphorylation, it can be turned off by dephosphorylation, and it can also be turned off by ubiquitin-mediated proteolysis, degradation of the cyclin subunit. So we now know, through the work of many, many groups, once the door had opened a crack, people rushed in, and the cell cycle field became extremely crowded, just the kind of place where Avram said you shouldn't be, because it's far too many people, far too competitive, and far, really clever people too, which makes, when you're up against clever, hard-working people, this is very bad news indeed. Because somehow they seem to get all the luck. I don't know what it is. So MPF turns on here and catalyzes entry into mitosis. Then something happens, and the second half of the talk is going to be what happens here, and you come out of mitosis. And then it turns out this is such a good invention, turning on protein kinases this way, but it's also used here to initiate chromosome replication. So this combination of a kinase subunit with a cyclin companion coming and going is used is not just a one-shot deal here, it's also used here, and probably also over here, if to determine the difference between cells resting and growing. Now, and get to the crunch. So as far as we know, these enzymes, protein kinases, cyclin-dependent protein kinase, CDKs, can only do something by phosphorylating target proteins. They don't have any other way of influencing things. So the question then becomes, when a cell enters mitosis, very simple, how many proteins have to be phosphorylated, how much, in order to bring about these dramatic structural changes in the behavior of the cell? And this has been a question right from the word go, right from about 1987 or 1988. And it's not a very easy question to answer, it turns out. And I'm going to show you our miserable failure to try and answer this question. So how do you answer this question? Well, now we get really biochemical. The first thing is to make your enzyme. And that we can do quite easily. It's very easy. Here is our enzyme, and you can see the two subunits. Cyclin A is the upper band, cyclin B is the lower band. So we can purify this stuff from bacteria, and we can purify it in a way that it has nice protein kinase activity. This stuff will, if you give it the precursor ATP, it will stick phosphates all over recipient proteins. So now all we have to do is to ask it, which proteins do you like to phosphorylate? So you go up to the protein, you say, so it won't tell you. So you have to do experiments to find out. And in our experience, this is the best way to do the experiment. Now, this gets really technical. But basically, what you do is you take lots of proteins, here revealed again as bands on gels, and you add the enzyme, and this is the control. I'm very keen on controls. They're really important. And here is a substrate, and the substrate responds by shifting slightly. It slows down its mobility on this gel. That is a substrate. So Todd Duncan was the first graduate student in my lab to do this, and this is one of his experiments, and you can see that Todd was a very good graduate student because his gels are beautiful. And every so often you can see, I mean, it's just encouraging enough, and boy, I'm having a hard time seeing it. I think somewhere over here is one, and somewhere over there is another one. Basically, this is one experiment, and if you're lucky, one protein shifts on this, and the hit rate is about 1%. And then after that comes some postdocs, Krupa and Alessia, and what we've done now is to make a sort of list of substrates, and this is sort of out of focus, and it doesn't really matter anyway, because these lists, this is called omics, this is proteomics or phosphoproteomics. It's actually not terribly enlightening, I must say, and what you have to do is to focus, and Alessia's been focusing on this one protein here called tipin, which actually controls something to do with DNA replication and DNA damage responses, because that's what interests her. And the net result of all of this, although we have a list now of over 100 proteins which get phosphorylated by these enzymes, it really doesn't help explain how these enzymes make the cells enter mitosis. And Sidney Brenner had a very nice thing of this omics thing. He called it low input, high throughput, no output research. And I must say I'm rather inclined to agree with him. But, you know, what can you do? You just have to sort of carry on and do something. So these are some more cells. These are human cells dividing on a plate. And you can see, again, the kind of problem. Suddenly a cell will round up. It divides. It's hard to see, but if you look very closely, if I were to play this movie several times, you would actually be able to see the chromosome jiggling around in the middle of the cell. And then they settle back down again. And recently we've become really interested in the question of settling back down again, getting out of mitosis. And it's exactly the same as the previous thing. It's seeing funny behavior of bands on gels. Avram referred to sort of the Pasteurian thing of luck favoring the chance favoring the prepared mind. So let's just think about this. Mitosis going back to interphase. So now the cell has got to grow to twice its size and then it can divide again. Okay? Well, I've said that protein kinases, which stick phosphate groups on other proteins, are what get you into mitosis. So it follows, as the night follows day, that getting out of mitosis must just mean taking those phosphate groups off. And funnily enough, that's a subject which has received rather little attention. And part of the reason is because somehow undoing stuff is not as interesting as doing stuff. You know, it's sort of like tidying up after you've done something. So it follows that there must be enzymes, phosphatases, these are the enzymes that take off phosphate groups, must restore the proteins to their interphase state to undo the CDK's work. And the hero of this story is just one postdoc who came from Japan, Sitaram Yashida, who is an extremely gifted young scientist. And he's what his former mentor, Mitsuhiro Yanagida, calls an ordinary person. Actually, he's quite extraordinary. But he's just very down-to-earth and practical, and he loves doing experiments. He's really, really good at it. And he worked, for example, in a soybean brewing company for a while and didn't like it. He wanted to get back into basic research because the business of soybean, there wasn't much of a challenge. And he's really, really good. Also very important in this research are these eggs, and these are the two people who do more than anything to ensure that our frogs are well kept and give rise to good eggs. Here on the left and Jane on the right. And here are the product of those eggs. I've shown you live eggs so far, but what we do is really rather cruel to the eggs. We crush them mercilessly. All you have to do is put them in one of these centrifuge tubes and spin them, and what happens is that all the black stuff you saw in the previous movie goes down to the bottom. Here it's been removed, and this is a supernatant. And what you're looking at here really is pure cytoplasm with nothing added and nothing taken away except the rubbish. And with this pure cytoplasm, you can do the most beautiful experiments because you can add things and subtract things without the nuisance of having to use micropipettes to attack the cells. So you can get lots of this stuff. Moreover, you can freeze it away in liquid nitrogen, and then you can do an experiment any day of the night. You don't need a fresh female frog. You can just do the experiment. So let's go back to this diagram of the comings and goings of MPF. And you may have noticed that in the middle of here was a very long period where MPF stays high. And there's a very good reason for that. In frog reproductive physiology, the egg actually stays in metaphase with the chromosomes all lined up until the sperm arrives. And this arrest is maintained by a factor which was also discovered by Mazui in his initial 1971 work. It's called cytostatic factor because it blocks the progress of the cell cycle. What's the difference between MPF and CSF? Actually, the only difference really is the assay. If you suck cytoplasm out of an egg and inject it into an oocyte, so this is pushing it back in time, so to speak, it initiates this autocatalytic loop that turns on the formation of CDC2 cyclin and or MPF and causes acceleration of the cell cycle. But if you inject the same cytoplasm into a fertilized egg, which is going to move forward of its own accord, what happens is a cell cycle arrest, and the cell arrests in metaphase with chromosomes lined up on this mitotic spindle. So what is this mysterious factor CSF? Again, it's heat labile, it's protease sensitive, it's clearly some kind of a factor or an enzyme. It's made of protein. And it's taken a while to find out what it was, and this is an example of how complicated a biological switch can be, going back to the second slide. So the first thing that was discovered that an oncogene, which is only found normally in eggs, called CMOS, was CSF. And then it turned out that CMOS, which itself was a protein kinase, was actually a protein kinase that activated other protein kinases, a protein kinase cascade called the MAP kinase pathway. And then it turned out that it wasn't really MAP kinase, which was thought to be the end of that pathway, but a downstream kinase called P90 RISC. So this is getting terribly complicated. Here it is. MOS turns on MEC, turns on MAP kinase, turns on P90 RISC. And then the target was discovered by accident, first by Peter Jackson and then by Thomas Meyer. And it turns out there is a protein which gets phosphorylated by this enzyme. A phosphate gets stuck on it, and that becomes a potent inhibitor of the degradation of cyclin. Poof! So then how does calcium get you out of this arrest situation? It's no good being arrested. You've also got to impose the arrest, and then you've got to be able to take the arrest off when the sperm arrives. So in all cases that I know of, when the sperm hits an egg, and this is true for frogs, starfish, sea urchins, whoever you are, probably even plants, when the sperm hits the egg, there's a huge increase in the calcium inside the cell. Now, how does that calcium work? Well, calcium turns on another enzyme, and by now it shouldn't surprise you to learn that the enzyme that turns on is a protein kinase. And it turns out that the calcium-activated protein kinase phosphorylates the inhibitor. Now, again, it's sort of ridiculously, stupidly complicated, but this is how biological, biochemical switches work. That protein kinase makes a target site for another protein kinase, which is called polo, for another jokey reason, which I won't go into. And that phosphorylation then makes this protein a target for the ubiquitin ligase system. We keep coming back to ubiquitin. And it's degraded, and once the inhibitor is gone, of course, everything can go forward. So very simply, so the cyclins are then degraded, also by ubiquitin-mediated proteolysis. So it's a kind of two-step process, and CDK activity is thereby turned off, and the cell cycle can go on, and MPF will go down. And here's the evidence, really, the most compelling evidence that is the case. In this experiment here, we're looking at cyclin, and we're looking at its disappearance. So you can see cyclin here disappears when you add calcium. If you don't add the calcium, it doesn't disappear. And if you add a form of the enzyme, the calcium-activated enzyme, which has been genetically engineered so it's active even without the calcium, then that itself will make the cyclin disappear. So QED, that's all you need. You just need to somehow turn on this enzyme and the cyclin will go away. And for many years, people thought, aha, that's it, problem solved. And then along came Satori. Now, what Satori's project was to investigate the mechanism of action of this interesting inhibitor of the APC. And this is a very competitive project because Avram is working on it, and we don't really want to compete with one another. Avram is a much better biochemist than I am, I must say. Much better. Satori made this reagent. It's a very nice reagent. You can make it and you can purify it very easily because it has a purification tag on it. And the question was, how does it work? So Satori added into one of these frog egg extracts, and now we get really scientific, so bear with me. Here's adding the calcium. The cyclin goes away. And we now add a new dimension into the assay, which is we look at the state of phosphorylation of one of the subunits of the ubiquitin ligase that's responsible for degrading the cyclin. Now, this is just a marker, don't worry about it, but basically, if it's high, it's got phosphates on it, and if it's low, it hasn't got phosphates on it. Okay? And what you can see is that when you add calcium, what happens is the cyclin gets degraded, the protein kinase turns off, and the APC gets dephosphorylated. Fine. So Satori came to me and he said, look, the reagent works perfectly because when I add calcium in the presence of this indestructible ERP1, the calcium can no longer work because we've removed the degradation signal from it. It can't be degraded. And he was really, really pleased. And I said to him, Satori, what's going on here? And he said, I don't know. That always happens, but I don't know. So what you can see is, and what caught our attention was that the APC, this subunit, is transiently dephosphorylated. The phosphates are taken off, and then they're put back on again. And we looked at that and we thought, gosh, that's very interesting. What the hell is going on here? And we thought, well, there's only one thing that's happening. Calcium is clearly turning on a phosphatase, which is transiently taking off. And as the calcium goes away, because it's sucked away into little vesicles in these very crude extracts, the kinases win and the thing gets rephosphorylated. So was that actually true? Could be true because it's well known that calcium, in addition to turning on a protein kinase, also turns on an opposing enzyme which removes the phosphate. Now, presumably you don't take off the same phosphates that you put on because that's kind of pointless. You just get a futile cycle then. But nobody had thought about this, so we thought about it and followed our noses, so to speak. And the first thing was that this enzyme, the calcium-activated phosphatase, indeed is turned on. And you can see when the calcium is high, it's high, and as the calcium is pumped away, it goes down again. Huge peak of activity, very sharp, almost all over by about 5 minutes. And luckily for us, there's a very powerful and specific inhibitor, which is used in kidney transplants, because this enzyme is very important to allow for the immune response, and if you inhibit it highly specifically with this drug called cyclosporin A, you can eliminate that peak. So then it was very easy to do the experiment. What happens if we don't have this peak of calcium-activated phosphatase activity? And the answer was sort of clear and yet not clear. First of all, if you forget these left-hand sides are controls, but here's adding calcium. You can see the APC getting dephosphorylated, the cyclin getting degraded. When you inhibit that calcium-activated phosphatase, there's absolutely no sign whatsoever of dephosphorylation of the APC. So experiment has worked. But we were kind of disappointed by this result over here, because we kind of hoped that it might have some really profound effect on the disappearance of the cyclin, and it just slowed it down. So that was irritating. But you always know when you're on the right lines in science, because nature is never quite the way that you predict that she will be. She always tells you something when you ask her a question. She tells you something that you didn't know before. So this was good. It sort of kept us honest. And then we began to ask other questions, where we've only so far been looking about the dephosphorylation of one particular subunit of one particular system in the cell. What about other things which get phosphorylated during mitosis? And if you look here, this is an assay which measures these black things here are all the phosphorylated proteins in this extract, in a mitotic extract. And this is what happens when you add the calcium. All this blackness disappears. Those are all the phosphate groups disappearing off those proteins. What happens if you inhibit this calcium-induced phosphatase? The answer is the blackness stays. So the calcium flash is really important for getting things out of mitosis. You can't, if you inhibit that phosphatase, you can't get out of mitosis. So then we did a more physiological assay, and basically what this physiological assay says that if you don't have the calcium flash, getting out of mitosis is not totally inhibited, but it's seriously delayed. This measuring it on the right-hand side here shows us a delay of about an hour in getting out of mitosis when you inhibit that phosphatase. And what was really nice, and this is a brilliant piece of work by Satori, he was then able to add this special calcium-independent kinase and a calcium-independent phosphatase, and showed that when he added the two together, so now that you have no calcium, but you've restored both the kinase and the phosphatase, then you more or less eliminate that lag. So we were very happy about that. But then we got concerned, because this is all to do with frog eggs, right? And I mean, frog eggs are interesting and they're fun and they're nice to work on, but they're not the same as human cells dividing. So we then asked whether you needed this calcium-induced phosphatase in real cell cycles, real mitotic cell cycles, and I was very pleased when we discovered, when we added the cyclosporin inhibitor, which would have totally blocked the eggs in M phase, it had absolutely no effect on the mitotic cycles. Here you see cyclin coming and going with exactly the same kinetics in the control and the experiment. So you don't need calcium for normal cell divisions. But you still dephosphorylate things. So that means there must be another phosphatase present. So at this point we decided we better look for some other phosphatases. And this sort of goes along with another question which bothered me right from the word go. How do you convert a continuous increase in something into a sharp switch-like behavior? Because cyclin, which is going to turn on the CDK, just accumulates steadily. And it's not at all clear what the nature of the threshold is that gives you this sharp increase. And doing experiments on this, again, in these crude frog extracts, it made us worry even more about this. Here you see cyclin accumulating, and here you see an assay of the protein kinase activity. The blacker this line is here, the more phosphates are being stuck on proteins. And you can see that actually the activity also goes up steadily, and yet you can see when the extract enters mitosis because of this sudden hyperphosphorylation of these mitotic proteins. So we began to think, gosh, there must be a phosphatase here which switches off in order to allow the cells. It's not enough just to turn on the kinase. You've got to switch off the phosphatase as well. But what is that phosphatase? Does it even exist, or are we completely wrong? So what we did was to build an assay, and the assay is a very simple one. You take a soluble bacterial protein, you stick a frog protein on the back end. The frog protein has a phosphorylation site on it, there's a serine there, which can be phosphorylated by our recombinant enzyme. So we can get a radioactively labeled phosphatase substrate, which we can add into these very crude extracts and see how they behave. And now we got another total surprise. It turned out that this substrate here, which is serine 50 of protein called CD13, which again is very familiar to Avram, and is actually phosphorylated in these extracts normally. This phosphate is removed by the calcium flash, and then as the calcium-activated phosphatase goes away, it goes down, and then, most unexpectedly, it comes back up again. And that coming back up again, you can prove is a different activity from the first activity. So we're dealing, it turned out, we were really shocked to discover this, not with one phosphatase, but with two phosphatases. The first phosphatase is activated by calcium and is wiped out by cyclosporine. The second phosphatase has nothing to do with calcium and is not wiped out by cyclosporine, is wiped out by an inhibitor that you find in shellfish poisoning called okadaic acid. So basically, one phosphatase leads to the appearance of another phosphatase. Does that phosphatase has got anything to do? We've shown that the calcium-activated phosphatase doesn't turn on and off in mitosis. What about this phosphatase? The answer is yes. This phosphatase turns off completely. When the extract enters mitosis, the phosphatase activity goes away. When it comes back out of mitosis, this phosphatase activity. This has exactly the properties we want. You turn it off to enter mitosis. You turn it off to get out the other side. Wonderful. But what is it? Well, we call it phosphatase X, and we call it phosphatase X for a good reason. We don't really know what phosphatase X is. Nor do we really know how it's regulated. So now this is where you're absolutely bang up to date. This is where we are. As usual in science, you come to a point where you rush forward and then you're stuck again. So we're in the middle of a thick fog at the moment. But I can tell you what we know, and it's puzzling. First of all, the evidence strongly suggests that phosphatase X, this is quite recent evidence, we got about a month ago, is in fact some form of an enzyme which is well known called PP2A. Now, unfortunately, protein phosphatase 2A, there are about 7 of them altogether, and PP2A itself has about 80 different forms, and we don't know which one we're dealing with, because although all these forms have one of 2 catalytic subunits and one of 2 A subunit, which is a kind of this thing down below is a regulatory subunit. This guy here, the yellow guy, comes in something like 16 different forms. And we don't know which one we're dealing with. We're working hard on that. So, nor do we know how this thing is turned on and off. We know a little bit about it because we know we can turn it on simply by inhibiting CDK activity. So that implies actually that the phosphatase itself is regulated by phosphorylation. And if that sounds a bit incestuous, it is. And moreover, it implies that there must be a phosphatase Y, because if it's really regulated by phosphorylation, negatively regulated by phosphorylation, it must be turned on by another phosphatase. So there are kinases that attack kinases, and there are phosphatases that attack phosphatases. But we don't know. We know much less about phosphatase Y than we know about phosphatase X. And the other really irritating thing about this makes it exceptionally difficult to study. The minute you dilute those very concentrated straw-colored extracts that I showed you, the phosphatase activity turns on. You can get about a 100-fold increase by a 10-fold dilution of those extracts, and we have absolutely no idea why and what that represents. It's something dissociating, something, it makes it very hard to discover because we can't work on the inactive form of the enzyme. So this is really quite tricky stuff. This also explains why nobody had spotted this before, because you don't see this behavior of that phosphatase unless you use these very, very crude extracts. If you did a standard biochemical assay, you wouldn't notice any difference in the phosphatase activity between mitotic and interphase cells. So here we are. This is the control pathway, and this is really this is the end. This is very simple. The CDK phosphorylates proteins. The proteins get phosphorylated. They change their behavior and leads up to metaphase in the cell cycle. And because the CDK inhibits the phosphatase, directly or indirectly, it's very unlikely that this line, I suspect, is a direct one. It means that the default state of the cell, once you have CDKs, is in mitosis, and you're stuck in mitosis, which means in turn that the only way of extricating yourself from this is by destroying the cyclin by the ubiquitin-mediated pathway and killing this CDK. Otherwise, you're locked in this state forever. And we don't know, as I said, really what phosphatase X is, nor do we know how it's regulated, and that's what we'd like to know. And it's really, really fun to have a problem which actually engages you. I mean, you always think that you're never going to find out anything ever again, and it's only fun discovering things. And this is a sort of typical voyage of discovering. After a long period where we really didn't find out anything new, we suddenly find a whole brand new field which nobody knew anything about before, and that's very good. So in conclusion, this is the message. It's really simple. If you want to fill a basin with water, you need to put the plug in. It's no good just turning on the tap, okay? And if you put the plug in, then the basin will fill up with water. So very simple biochemistry. Thank you very much for listening.